I have a story to tell you today that is the story of the discovery of the structure of the molecule of DNA. And as Sandra said, it's a story of strong personalities, of drama, and I think some important lessons for interdependence. Our story has a cast of characters. Rosalyn Franklin is the heroine, and like any good heroine, she will die a tragic death. James Watson, on the left, is the villain today. And he's a villain who believes himself to be the hero. James Crick, on the right, is his foil, who will eventually befriend the heroine. And our final major character is Wilkins, their stooge, Maurice Wilkins, who never quite seems to understand what he did or what he did wrong. So the story. Oh, excuse me, one other character, the DNA structure, and this is a model that is a replica, or it's actually reconstruction of the model that Watson and Crick built. And it has been reconstructed using the pieces that they used, and now is in the Science Museum in London. So, our story. So Rosalind Elsie Franklin was born the 25th of July in 1920. Her parents were Ellis Franklin and Muriel Whaley Franklin, and they lived in the western edge of Bayswater at London W2, if you're familiar with London, now known better as the fashionable Notting Hill district. They were a strong, traditional Anglo-Jewish family. They were part of the cousinhood, called that because of the number of intermarriages in the group. They were outstanding citizens. They were publishers, owners of publishing houses, bankers, and outstanding philanthropists, especially working for the education of the working class. So Rosalind Franklin, as an adult, was an English gentlewoman and a Jew. She was also called by an aunt as alarmingly clever. She was the brightest and the most determined of her siblings, and she was closest to her three brothers, and she joined them in most of their activities. She was especially good at sports and at science. So in January of 1932, she entered St. Paul's Girls' School, which is an academically rigorous London day school, which is still open today and still prides itself in incorporating girls from all walks of life who seek real intellectual stimulation. So they open admission to children of all faith and of no faith. By the age of 16, she took science as her subject, and she was especially strong. She had especially strong interest in chemistry, in physics, mathematics, pure and applied. And the school had relatively modern science facilities, but she had little exposure to experimental work. 5 months before her 18th birthday, which is fairly young, she sat for the entrance exams in physics and chemistry at Cambridge. Although the people at the school wanted her to wait another year, she was ready to go. And she was offered places at both women's colleges in Cambridge, Girton and Newnham, and she chose Newnham and started that fall. Oops, sorry. Which was a year earlier than most of her class. She earned an award, as a result of that examination, she earned an award of £30 per year for 3 years. But because her family was well enough to do, she accepted the honor, but did not accept the money. This was something that was done traditionally. And interestingly enough, Penn State is doing something similar this year. In fact, they say they thought of it, but clearly. So she received that award because of all of the people who took the test that year. She had come in first in the Cambridge examination in chemistry. So Cambridge at that time began admitting women in 1869, and they began admitting Jews in 1871. But at the time she entered, they didn't recognize women as members of the university. They were students of Girton and Newman colleges, And instead of being granted degrees, they received degrees titular, which, of course, the boys like to call degrees tit. There was a quota of 500 women, and they were admitted to the men's lectures, but they were expected to all sit together. In 1948, after Rosalind had graduated, Cambridge recognized women as members of the university and conferred degrees retroactively to women, including Rosalind. So while she was there, she worked very hard at her courses, including chemistry, physics, mathematics, mineralogy, scientific German, and she learned the basics of a technique called X-ray crystallography. So in X-ray crystallography, what happens is an X-ray source is used instead of light, so it's a form of photography, except it's X-rays instead, and when the X-rays shine through a crystal, the atoms diffract or scatter in a particular direction, and they leave spots on a photographic plate that's caused by the scattering of the X-rays when they hit the atoms of the crystal. So those spots vary in intensity in the X-rays, and they can reinforce each other or cancel each other out, and that leaves a pattern. So in an X-ray photograph, what you see is a diffraction pattern caused by the interaction of the X-rays with the crystalline lattice. So you're not seeing an image of the molecule, you're seeing how it's reacting to being hit with X-rays. It turns out that this is the technique that Rosalind became so expert at, and it was her work in X-ray diffraction that was her major contribution to the understanding of the DNA molecule. While she was doing all of this, she also worked hard at sports. She went from hockey to squash, boating on the Cam, tennis, long bike rides. In fact, she rode her bike from Cambridge back to London. And during the war, more women became more prominent in academics and industry as the proportion of women at Cambridge increased and as more men were involved in military service. In October of 1940, she entered her third and final undergraduate year, and scientists and other male faculty were focused on the war. That term, she met a woman named Adrian, I'm not saying this right, Adrian File, who was an eminent French physicist, but she was in London. She had been a pupil of Marie Curie, and she was also Jewish, and she had left France in response to de Gaulle's call for the French people to join him in England. Meeting Adrian File was a turning point in Rosalyn Franklin's life. She finally met a professional scientist. Early in 1941, the Blitz had intensified. Rosalind was anxious about her exams. She was sick. She had taken some medicine to help her sleep. And so her exams didn't go as well as she had hoped. She achieved a good second, but she was top on the physical chemistry exam. And for that, she received an additional scholarship of £15 to study another year, and she also received a research grant from the Department of Scientific and Industrial Research. Her first research project, however, was pretty trivial and it was disappointing, and she was frustrated that she wasn't given work related to the war. By the end of the summer, she was now 22, she moved into a hostel that was run by Adrian File, who was working at the Cavendish Laboratory, which is the great biophysics laboratory at Cambridge. At that point, Rosalind needed to decide whether to stay at Cambridge for PhD work or to seek a government job, or she might be called into military service. So in August of 1942, she accepted the opportunity to do research in a government lab in London, working for the British Coal Utilization Research Association called BCURA. And there she studied coal and charcoal because of their use in gas masks. So she tested changes in the porosity of coal under various conditions and temperatures, doing very careful experimental work, and eventually gained an international reputation for her work on coal. During that time, she was able to visit Paris. She had visited it once before. She visited Paris after the liberation in 1944, and she loved going abroad. By 1945, she received her PhD in physical chemistry, and her first scientific paper was submitted for publication. That summer, she went on a vacation to the Alps and stopped in Paris to visit Adrian File. And there she met some of Adrian's co-workers as well. So that the following fall, the French crystallographers Jacques Mering and Marcel Mathieu, who were friends of File, visited Rosalyn in London at Adrian's suggestion. And she seemed especially to be friends, Rosalyn seemed to her friends to be strongly attracted to Mering, who unfortunately already had a wife and a mistress, so he was kind of busy. But within a few weeks, she had an offer to study physical chemistry of coal in Paris. So she had a job offer. So in February 1947, she moved to Paris to work in a French government lab on crystallography. They called the lab the Labo. It was the Laboratoire Central des Services Chimiques. I don't speak French. Lo siento, in the 4th arrondissement, and her position was one of 15 researchers under Mering's direction. He taught her how to use X-ray diffraction. He helped her really learn that technique to study the internal structure of charcoal and clay. Other colleagues included the Italian crystallographer Vittorio Lussazzi, who was married to an Argentinian physician and who had spent his time during the war in Argentina. He was also Jewish. And that was the beginning of a very long and close friendship. And one thing you'll notice is that the next series of photos that I'm going to show you are all photos of Rosalyn during this time in her life, which has been considered to be the sort of happiest time in her life. She was part of a very active, very vibrant lab group. This is their tradition of having coffee in little crucibles in the lab. So she's not, this is part of the social aspect of the laboratory, was to have coffee in the middle of the day. She also, because she was there living in Europe, she was able to travel much more, go on holidays. She loved hiking. These are her favorite Norwegian hiking boots. She would go climbing. They would go as groups together. She would go with the Merings. She would go with the Lussatzis. And these photographs are, I don't know about this one, but the other photographs are all taken by Vittorio. So they were very good friends. Here she is looking out on the Tuscan landscape. Here she is in a French cafe, and this is a relatively famous photo of her sitting. It's a very intimate photo of a very happy young woman who also happened to be a very productive scientist. So she enjoyed the work, she enjoyed the fellowship of the Labo and the food and the fashion after living in wartime England. She enjoyed all of these walking trips, especially to the Alps. And she was very productive in her research and produced a series of important papers in top journals. Also in Paris, she became good friends with Anne Serre, whose husband was the American crystallographer David Serre. And it's Anne Serre who wrote the biography, the first biography of Rosalyn Franklin, who helped draw attention to Rosalyn's story. As she was gaining international reputation for her research, at the same time, she was getting pressure from her family to return to England. She would travel back and forth, but they wanted her home. And so she began looking and applying for positions and applying for funding. And in 1950, she received a Turner and Newell Research Fellowship to work for 3 years at King's College London under J.T. Randall to work on proteins in solution. So here's John Randall. Randall was the head of King's physics and biophysics programs, and King's is one college within London University. Rosalyn was being so productive in Paris, she requested to postpone her departure for a year, and in the meantime, interest in Randall's group shifted from proteins to molecules called nucleic acids. And they were interested in those especially because they had received a very nice preparation of DNA, a nucleic acid, from a worker in Switzerland that had been given to them and then brought back to study by Maurice Wilkins, who was in this lab. So we have Randall at the head, and Maurice Wilkins is starting to do some X-ray crystallography on these samples of DNA from Switzerland. During that time, Rosalyn received a letter from Randall that completely changed the direction of her research. This is the original letter, and I'm going to read you a small excerpt. He says, after very careful consideration and discussion with the senior people concerned, presumably Wilkins, it now seems that it would be a good deal more important for you to investigate the structure of certain biological fibers in which we are interested. By that he means these fibers of DNA, so they could take the solution of DNA, pull out small samples of it, pull it, and draw it out into fibers. And then they could take those fibers and use them in an X-ray machine, both by low and high angle diffractions, rather than to continue with the original project of work on solutions, that is, protein solutions, as the major one. This means that as far as the experimental X-ray effort is concerned, there will be at the moment only yourself and Gosling. Gosling is a graduate student she was given to supervise his PhD. The letter goes on to say, Gosling, working in conjunction with Wilkins, has already found that fibers of desoxyribose nucleic acid, that's DNA, an old name for it, derived from material provided from Switzerland, give remarkably good fiber diagrams. Now, when Rosalind received that letter, she was confused, she was surprised. She wasn't a biologist anyway, she was a physical chemist. She didn't know very much about proteins. She certainly knew nothing about nucleic acids. But she had a job, she knew that she was going to apply this technique, and she was ready to do that. One of the most interesting things about this letter is that Wilkins, apparently, who had been doing this work, didn't know that it had been sent. It was his understanding that Rosalind was coming to work with him as an associate on DNA, whereas this letter implies that Wilkins is not going to be working on DNA anymore. The existence of this letter apparently was not known by Wilkins until it was discovered in Rosalind's papers after her death. So, as I said, this stuff called desoxyribose nucleic acid was an older name that was used for DNA. And it was known that the heritable units, or genes, were strung like beads along chromosomes. But it wasn't understood how that information from the chromosomes was transmitted from one generation to another. Chromosomes can have two components, protein and DNA. Proteins are very diverse, they're very large, they're very complex molecules, whereas DNA superficially seems like a very simple molecule. It has only 4 kinds of repeating units, and so it seemed too simple to carry on the complex information of inheritance. So most of the research focus at the time was on proteins, and it was believed that that's where the key was going to be to understanding the secrets of inheritance. In 1943, Oswald Avery, an American, and his co-workers had demonstrated, however, that DNA was the transforming principle. But his work was largely ignored, and he was not I'm going to go tell them all what's right. And so most people didn't pay much attention to what he had done. The other thing that had happened was in 1949, Erwin Chargaff had analyzed proportions of these 4 bases, and they had found that the numbers of the molecules present of 2 different bases, adenine and guanine, I'll show you those in a minute, called purines, were always equal to the total number of thymine and cytosines, the other 2 bases. Let me just go ahead here for a second. There we go. Okay, so this is the science part of this talk. So DNA consists of a sugar called deoxyribose, which is a ring with an oxygen on it. Okay, that's a sugar. Doesn't look very sweet, I guess. It also has a phosphate group. So here's this sugar. There's a phosphate group. And then it has one of 4 nitrogenous bases. And in DNA, those nitrogenous bases are 2 of them are single circles, so cytosine and thymine, which are referred to as pyrimidines, and they're both a single ring. And then there are 2 purines, which have double rings, adenine and guanine. And so what Chargaff had found was that adenine and guanine which are always equal in total amount to the amounts of thymine and cytosine. And that was seen as a really profound thing, but nobody knew why. Okay? It was a really important piece of the puzzle. So today we know that DNA consists of 2 chains of nucleotides that are wrapped around each other in a helix. And we know that each of those chains has a sugar and phosphate, a sugar and phosphate, sugar and phosphate, sugar and phosphate. And then the bases, out here, the bases are the yellow. So this backbone is sugar, phosphate, sugar, phosphate, sugar, phosphate along the outside. And then these bases are in the inside. Okay. And here's another more detailed picture of them. So this is the way they would connect with guanine, cytosine, adenine, thymine. But it isn't just a single chain. In fact, there are 2 chains, and those chains are caused by the bonding together of adenine with thymine, always, so A with T, and guanine with cytosine, G with C. And you notice that in these pairings, that means that you have a pyrimidine and a purine, a pyrimidine and a purine. That is a double ring with a single ring, a double ring with a single ring, so that these look more or less, if you squint, kind of the same. They look very similar in size. The other thing that's important that you'll need to know about DNA is that, so here we have one sugar, phosphate, sugar, phosphate, sugar, phosphate going up one side of the ladder with that base stuck out in the middle. The opposite chain is oriented in the other direction. It's what's called antiparallel. So one is going one way, the other is going that way, like this, okay, with a hydrogen bond in between. So we have sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, okay? So that the 2 chains are almost standing on their head next to each other. So now I need about 10 volunteers. And we're going to do what I call in my classes the replication robot. Music, please, yes. I need 10 volunteers. I need 10 volunteers. That means people need to get up. Okay, so I want 5 of you to make a line facing this way. Stand this way. And I need 5 of you to make a line facing this way. So turn around this way. Right? Yep. Got it? Okay, now. So each of you, that's okay, as long as I have 2 people. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Good. Okay. Now, so each of these people is one nucleotide. Can you hear me? Yeah. Yes? Okay. So each of these people is one nucleotide. Their body is the sugar. Their left hand, is this right? Oh, you guys did it wrong. Turn around this way. You guys turn around this way. Good. Their left hand is their phosphate group. They're going to make a phosphate sugar chain by putting their hands on each other's left shoulder. That's it. Okay, so now we have the 2 chains. Then their right hands are their bases. And now if they join right hands in the middle, oops, we're off. We're off. Got it? Good, because otherwise we need some enzymes to come in here. Here you are. So you see the antiparallel chains that the two backbones are made of these phosphate, sugar, that they're oriented in different positions, and that the 2 complementary bases are joined by a hydrogen bond down the middle. Now the really neat thing, and you'll see this when we get into the structure of DNA, is what that means is that for each side there is only one unique partner. And when I come in as an enzyme and I start to break those bonds, new nucleotides can come in, and each chain is serving as a template for an exact sequence of complementary bases. And that's how DNA is able to reproduce itself so precisely, generation after generation. Thank you. There! There you go. All right. So, back to Rosalind. Where was I? Okay. So, in January of 1951, Rosalind moved to London from Paris. London in 1951 was still recovering from the war. The city was grim. Food was not very readily available, at least not high-quality food. She lived with her parents at first when she returned, and she was used to living independently in Paris. And her accent betrayed her as an upper-middle-class woman. And that set her apart from many of her coworkers. Dr. Jean Hansen, one of the women in the lab in the biophysics unit where she went, said she looked like an aristocrat and she acted like one. King's College also is known mostly for its Department of Theology, and so women were not allowed, and so during the day there were many clergy going through. It's a very religious area. There's lots of songs and church bells ringing all day long. Women weren't allowed at the time in the King's senior common room where the staff ate lunch, so women couldn't eat with the men at lunch. Not that any kind of connections or networking goes on at lunches, of course. And there were many ex-military men. So as her biographer Brenda Maddox says, in short, an upper middle class Anglo-Jewish woman with French tastes in serious discourse suddenly found herself in an environment friendly to everything she was not. When she arrived, Randall introduced her to the staff, including Raymond Gosling, who was going to be her PhD student, Alex Stokes, a physicist and mathematician, and in fact, 8 of the 31 biophysics staff in total were women, and several of them were in high positions. But I think this picture gives you a pretty good idea of what that lab would have been like to be in. It's not a group that I would really want to have fun with, let's say, or collaborate with based on the pictures. Interestingly enough, when she arrived, the one person who was not there was Maurice Wilkins, who was Randall's assistant director of the biophysics unit. He was away on holiday, and he's the one, you remember, who had been working intensively on nucleic acid structure and using X-ray photographs to show that this X characteristic of a helix in his X-ray photographs, and the one who didn't see the letter that Rosalind, that implied that he was no longer working on DNA. Wilkins has suggested to Randall, so it was Wilkins himself who had suggested to Randall that Rosalind work on DNA, but as I say, he was unaware of the letter. Wilkins, who shows up in this picture in the middle of the picture, okay, with the glasses, drinking from the cup. No, he's not drinking. He's this one right here, there. He was a Cambridge graduate himself, and then he had gone and worked at Berkeley, California, at the Livermore Laboratory on the Manhattan Project. So he'd worked on the bomb. While he was there, he fell in love with a woman. She became pregnant. They got married. And then when he had the opportunity to return to England, she was not interested in returning. He was not interested in staying. And their marriage ended very quickly in divorce. And when he returned to England then, he says in his memoir, he was really intent on finding a wife and having a family. So Rosalind got there, and she set to work setting up a new X-ray apparatus and writing up her results from Paris. There we go. We'll go back to that. In May, Wilkins went over to a conference at the Marine Biological Station in Naples, Italy. This is a world-famous biological station. I think the first one really built for the purpose of looking at marine organisms right there in the communities. And Wilkins presented his X-ray images of DNA at this conference. In the audience was this fine-looking, handsome fellow, 23-year-old Dr. James Watson, who was an American, and he was doing his postdoctoral fellowship in Copenhagen. Now, Watson was already somewhat of a boy wonder. He went to the University of Chicago at age 15. He had his PhD at 23. Okay? But Watson was also a typical 23-year-old. Okay? He was looking for a date. He was looking for a good time. But he was also a brilliant scientist. And Watson was at this meeting with his sister, who apparently was quite pretty. So he decided that when he heard Wilkins talk and he saw his images of DNA, he realized that this is the thing to be working on. DNA is the most important thing to be working on, and that's what I'm going to do. And he set about to write letters and find a position for himself in Cambridge, in England, trying to work on X-rays. And he went to the point of even using his sister, who accompanied him to Naples, to get to know Wilkins. And he says himself, if Maurice really liked my sister, it was inevitable that I would become closely associated with his X-ray work on DNA. Meanwhile, back in London, Rosalind began working with the assistance of Gosling on refining the procedures she had used before to make X-rays images of DNA. She had a new camera that was based on what she used in Paris, and it took her months to get that camera working, to get all the parts together, to get everything machined. And one of the things she found was that she needed to control the humidity of the chamber in which the fiber was supported, and that she could reversibly hydrate and dehydrate the fibers of DNA. So add water, make it hydrated, and then dry it out a little bit, make it dehydrated. The relationship was clearly strained between Rosalind and Wilkins. Almost from the start, they did not get along well. They were temperamental opposites. She was extremely direct and confrontational. She thrived on active debate. If she was used to a lab in which if you disagreed with somebody, you talk about it. And that was a positive sort of interaction. Wilkins, on the other hand, was much slower. He avoided eye contact, and he avoided conflict. He was still recovering, apparently, from the pain of his broken marriage, and as I said, he was actively seeking female companionship. But Rosalind certainly did not seem interested in any romantic relationship of any sort. Meanwhile, in California, another character, Linus Pauling, one of the most famous chemists and most probably significant chemists who ever lived, had described a structure called the alpha helix, which is one of the basic units of most proteins. And he found this structure by building physical models. So here he is, and this is his model of an alpha helix. So an alpha helix is simply a single helix. It would be one of these strands is an alpha helix. And this molecule that I'm showing you here is the molecule collagen, which is the main component of, say, your Achilles tendon. It's what holds you together. And it consists of 3 alpha helices woven together. That's exceptional, but many proteins have regions in them that are alpha helices. So this was a major understanding to the structure of proteins. So Linus Pauling had already a world-renowned reputation for getting to the root of a problem, finding something very quickly, and moving on to another problem, finding that solution. As Francis Crick later said, because of this work, helices, the notion that molecules might be helices, was in the air. That year, at a conference at the Cavendish Lab, this lab in Cambridge, Wilkins discussed the X-ray work that King's was doing and showed an X-ray with a strong central X that led him to conclude that all of the DNA was helical. And Rosalind was surprised that he was talking about DNA and was quite angry and said to him, "Go back to your microscopes." Okay? Wilkins was shaken and he didn't understand. He hadn't seen Randall's letter to Rosalind. And she had assumed that Wilkins wasn't working on DNA. Meanwhile, while Rosalind was away, Wilkins wrote to his old acquaintance, Francis Crick, who was at the Cavendish Lab in Cambridge. And Crick, a bit older, he was about in his 30s at that time, had also worked in science labs during the war, and then was now going back to finish his PhD. So he was a bit older to be a PhD student. And he was supposed to be working on proteins because, of course, everybody had thought proteins was where it was at. But Wilkins was a friend of his and said to him, "You know, I think DNA is probably helical." When Rosalind returned from out of town, she continued to work on her X-ray diffraction, and one of the things she found is that there were two forms of DNA. Now, she wouldn't have had, this is a modern figure, but this gives you the idea of what it is. There's A DNA, which is what happens when DNA is drier. Okay? So the dehydrated is A. And then when she would put more water, when she would change the humidity and hydrate the DNA, she would get a very different diffraction pattern that turns out to be resulting from the B DNA. Now, B DNA, as we now know, is the form of DNA when DNA is about to replicate, when it's going to split, when it's going to loosen up. And the spacing of it is such that it gives a much sharper, clearer X-ray diffraction pattern than the more compacted A DNA. All of the work that had been done before was on the A DNA, and so all of Wilkins' X-ray images were kind of muddy, they weren't as clear, they didn't have as much information. But Rosalind, because she was able to control the humidity, was able to get truly amazing images of the B form. And she was the only one working on it. So there she was, busily working on this DNA that had come from Switzerland, and Wilkins had obtained additional DNA from Erwin Chargaff, the one with the ratio of A to D and C to G. And he suggested that so Wilkins suggested to Rosalind that new DNA might give useful images as well, and that they should collaborate. Well, she had it, and she said, "How dare you interpret my results?" That he was implying that everything should be helical. She saw no evidence at that point that it was helical, and she felt as if he was sticking his nose into her research. They reached an absolute standoff. They couldn't communicate to each other, and so Randall, the head of the lab, came in and arranged a compromise that Rosalind would continue to work on the A form using the really good DNA from Signer, and that Wilkins would only work on the B form using the DNA that he just got. Well, that was a horrible solution because Wilkins' DNA from Chargaff wouldn't crystallize, so he could never see the B form. And Rosalind, it turns out then, spent all her time working on the A form, which gave not as much information. Rosalind, in what she had seen, realized that the structure of the B form was helical, and also that the phosphates were on the outside of the helix. You remember the backbones, okay? And that was what made it possible for the molecule to take up water. During this time, Wilkins started visiting Cambridge more frequently. So we'd go up from London to Cambridge, especially to see Francis Crick and his wife, Odile, who was an excellent decorator and cook and an all-around lovely person. So he would go up and have a nice dinner and socialize with them. And Crick had started discussing and collaborating with James Watson. Watson had arranged a transfer finally to Cambridge from Copenhagen. And even though Crick was supposed to be finishing his PhD on the X-ray diffraction of proteins, Watson was so keen on this DNA stuff that he convinced him that really they should start working on DNA together, and they should do this by building models, because that's what, there's my mind. Pauling, thank you. Yeah, good. Just seeing if you were listening. Linus Pauling had used models, and so Watson decided, oh, this is the way to get at the answer. So once again, then in November, Crick invited Wilkins to Cambridge again, have dinner at his house, and he invited Watson. And the two of them pumped Wilkins for information about DNA. Wilkins had no idea that Watson and Crick were working on DNA, and they didn't let on, but they said, oh, you should use models, you should do this. And Wilkins mostly complained about how difficult Rosalyn Franklin was to work with. Rosalyn had no interest in building models because she came from a tradition in which you do careful experimentation, you collect data, and you use the data, the evidence, to drive what you do. And it's assumed that she saw model building as sort of an ad hoc way of approaching a problem, that you only started to build a model once you really understood the structure of the molecule. So that November, there was a colloquium at King's College in London. Wilkins came, and Wilkins spoke and repeated mostly what he had said earlier. Stokes was there, gave a summary of his helical theory for interpreting X-ray photographs, and Rosalyn gave a talk about the A to B transition in DNA. But and this is another very strange thing, her notes mention the possibility of a helical structure. But no one who attended that talk has any recollection of her mentioning helices. And so from that, the three men who had been talking together had decided that she was anti-helical and that she had totally misunderstood her photographs and couldn't see that this structure was helical. One person in the audience was our friend James Watson, who was anxious to hear Rosalind's findings. So Crick couldn't go, and it was Crick who really was the X-ray crystallographer. So he sent Watson, and Watson was there, and he was, primed to collect information. And he was also curious to see what she was like, because he had heard from Wilkins what a nasty person she was. And his recollection is this: momentarily, I wondered how she would look if she took off her glasses and did something novel with her hair. He took no notes, and he missed completely what she said about the phosphate groups being on the outside. So he went back to report to Crick and blundered the whole business. Rosalind at this point had had it. She began looking for opportunities to work elsewhere. But in the meantime, she began this extremely laborious set of calculations to analyze the A form of DNA. And sometime during this period, in a very long exposure, she produced this photograph of the B form, which is labeled 51, which shows an extremely sharp image. This X is what tells those who know what they're looking at that this is a helical molecule. The spacing of these bars indicates information about the dimensions of the molecule. But all of this needs to be gotten out of these images with a series of very complicated and lengthy calculations, all done, of course, by hand at that time. So she's working away, but she put this beautiful image aside because she was supposed to be working on the A form. And then she informed Randall that she was going to leave. She had received an opportunity to work at a different college in London, Birkbeck College, to work on viruses. Crick and Watson, because they had been fooling around with DNA, their boss told them, stop, you're supposed to be working on other things. And they had been told to leave DNA to King's College, not to work on DNA, and get on with what they were supposed to be doing. But they continued to think about it and to talk about it informally. So at the end of that year of 1952, each of Randall's groups again wrote reports, and Rosalind's report included for the Medical Research Council, which was the source of their funding. So they had a report that was an internal report to their funding source. And Rosalind's report included details from the data from her photographs. It just so happened that in Cambridge, Linus Pauling's son came to work at the same lab as Crick and Watson. And so they had an inside line on what Linus Pauling was doing in California. And they soon received a draft, his son received a draft, of a manuscript of Linus Pauling describing the structure of DNA. They took a look at it, and they immediately saw it was wrong, and they were delighted. But they also realized that he was going to get it right eventually, and that if they wanted to beat him to it, they needed to step on it. So once again, they called Wilkins down. They decided that since Rosalind was going to leave King's, then Wilkins was going to be the only one at King's working on DNA. So they got Wilkins' begrudging approval that Watson and Crick would go back to building models of DNA. During that time, Watson dropped in on Rosalind in her office. And he says, since the door was already ajar, I pushed it open to see her bending over a lighted box upon which lay an X-ray photograph she was measuring. Momentarily startled by my entry, she quickly regained her composure and, looking straight at my face, let her eyes tell me that uninvited guests should have the courtesy to knock. He offered to show her this draft of Pauling's paper, which was pretty obnoxious because she had been corresponding with Pauling and Pauling hadn't sent her a copy. Then he proceeded to tell her what was wrong with Pauling's paper. She replied that a helical structure still hadn't been proven. That is, she really was wanted to look at evidence. Watson goes on and says, "I was more aware of her data than she realized. Several months earlier, Maurice had told me the nature of her so-called anti-helical results. Since Francis had assured me that they were red herring, I decided to risk a full explosion. Without further hesitation, I implied that she was incompetent in interpreting X-ray pictures." He shows up at her office. "If only she would learn some theory, she would understand how her supposed anti-helical features arose from the minor distortions needed to pack regular helices into a crystallizing lattice." Suddenly, Rosie came from behind the lab bench that separated us and began moving towards me. Fearing that in her hot anger she might strike me. This is a woman about, this big. I grabbed up the Pauling manuscript and hastily retreated to the open door. Whew, he escaped her wrath. Rosalyn may have been angry for various reasons, not to mention he barged in on her work. But she had already complained to a colleague, Simon Altman, that she had found her notebooks being read and that she suspected that her desk had been gone through. She knew that Wilkins was friendly with Crick and Watson and was concerned that he was sharing her data with them. So, sometime in January, Gosling, her PhD student, showed Wilkins photograph 51. And that's understandable because Gosling was Rosalind's student. This was part of his work as well. He understood that she was leaving, and he showed Wilkins the photograph. But Wilkins then showed the photograph to Watson. There were many diffraction photographs of DNA around in the lab, and this one was the best one. So he showed it to him. Then the photo had a huge impact on Watson. He could clearly see the DNA was helical with a definite pattern of spacing. He says in the double helix, "The instant I saw the picture, my mouth fell open and my pulse began to race." On the train back to Cambridge, he sketched everything he could remember about the photo on the margin of his newspaper. And the next day, Crick and Watson went to their boss, and they were freed to work on DNA. They set to work immediately to interpret the information from the photo, and within 5 days started working on a new model. Watson was convinced that there were 2 strands to the helix, and Crick suggested that they put the phosphates on the outside. So they're almost done with the model. They invite Wilkins up again. "Have lunch, come have lunch." He also invited Watson, and Wilkins accepted, saying, "I will tell you all I can remember and scribble down from Rosalind." At the lunch, Wilkins agreed to start model building as soon as Rosalind had left King's, and he grudgingly agreed that Crick and Watson could start work again on DNA. They didn't even tell him that they were already building the model. The next week, Crick and Watson had another friend working on their side, apparently, Max Perutz, who was in their lab, and who was on the research council to which she had given her final report, gave them a copy of her report. Apparently, he didn't understand that perhaps, even though it wasn't marked confidential, because it was unpublished results, it isn't quite the thing to do to share data. Meanwhile, Rosalind had finished her analysis of A and began her analysis of B, the one in the image. And as soon as she started measuring this image, she realized that both forms, the A and B forms, were 2-chain helices. And she also began to appreciate the significance of the Chargaff ratios, but she didn't quite make the next step to base pairing. Watson had the help of another friend, another colleague, who showed him that he was using the wrong form of the bases and then realized that the AT and CG pairs would have similar shapes and dimensions as we saw earlier on. And so they continued to work on how the model would be built. On the 7th of March, 1953, Wilkins wrote to Crick, "I think you will be interested to know that our dark lady leaves us next week. I am now reasonably clear of other commitments and have started up a general offensive on nature's secret strongholds. At last the decks are clear and we can put all hands to the pumps. It won't be long now." He was too late. The model was finished that same day in Cambridge, and Crick was standing in front of it when he read the letter. They didn't even have the courage to let him know that it was done. They had a colleague let him know. They invited him down to see it. And they talked that day, amongst the three of them, whether Wilkins should be a co-author or not. And Wilkins decided, no, he wouldn't be a co-author. He'd write his own paper with theirs. But they didn't quite know how to cite the unpublished evidence that had aided them all along, which was Rosalind's. As it happened at the same time, Gosling and Rosalyn were finishing a manuscript of their own. So Randall, their boss, made sure that the two papers from King's would appear along with Watson and Crick in the same issue of the journal. And this is another really interesting part of this story. So all of these papers were published in a journal called Nature. This is a more recent one. It is seen as the hottest, newest, most important science. It comes out every week. It's a British publication. The papers in it are extremely short, and it's understood that people usually publish a more lengthy version of their paper. But the papers are all peer-reviewed. That is, when you submit a paper, they send it to your peers, to other scientists, who review your work, decide whether it's worthy of publication, and then give feedback back to the journal. That whole process takes quite a while, even today, where everything is done electronically. But somehow, the Watson and Crick paper appears first in this series of 3 papers. Then there's a paper by Wilkins, Stokes, and Wilson, so Wilkins and his co-workers. And then the 3rd paper in the series is Franklin and Gosling, even though really the Watson and Crick paper depend upon the evidence of the Franklin and Gosling paper. Normally today, what you would do is either cite unpublished evidence, personal communication, but of course there was no personal communication because Franklin never gave them her data and they never admitted that they had seen it. And this was handled by Watson and Crick saying, "We were not aware of the details of the results presented there in these other papers when we devised our structure," as if they were doing this in a vacuum. A year later, in another paper, they did concede that without this data from King's, the formulations of our structure would have been most unlikely, if not impossible. The other remarkable thing about this paper, here they are, celebrating with a cup of coffee. That's almost the entire paper, 1 page. There's a little paragraph on the 2nd page. They include 1 sketch. It's very general. It's a proposal for a model, but once people saw it, they knew that they had gotten it right. And the last sentence of the paper is what then really set the framework for all of molecular biology. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In other words, just as I broke our strand and then said that more people could come in, they saw that immediately, given the backbone and the complementary pairs, that all the DNA had to do was split down the middle and there would be templates for the 2 sides. Instead of her contribution to all of this being acknowledged, Rosalind was forbidden by Randall, her boss, from working on nucleic acids once she went to Birbeck's, once she left the lab, which of course you can't tell someone you're not allowed to do this, but it was done. At Birkbeck, oh, I'm sorry. So this is, just go back for a second. This is a rather famous photo of Watson and Crick. So here's Watson, 23, Crick, 12 years older. This is their model. There's a lot of irony in this. This was shot months later after the publication, Crick is holding a slide rule, which they didn't use at all for this. It's a completely staged photo. But they became immediately famous. I mean, it was in Time magazine. It was publicized all over. At Birkbeck, Rosalind took up crystallography to understand the structure of the tobacco mosaic virus. Because viruses consist only of protein and nucleic acid, they're extremely useful for understanding the basic underlying principles of molecular genetics. And so there was a lot of interest in, there continues to be a lot of interest in viruses. She continued to be extremely productive, especially in her collaborations with Aaron Klug, and she gained an international reputation in viral research. She was invited to give seminars and conferences, including several in the US. And on a trip to the United States in 1956, she began to experience sharp abdominal pains. As soon as she returned to England, she saw physicians, and on the 4th of November, she had the first of a series of surgeries for ovarian cancer. She continued to work and travel between bouts of chemotherapy for the next year and a half. And she died on the 16th of April, 1958. Her boss at the time, Randall, said of her in an obituary in Nature, "As a scientist, Miss Franklin was distinguished by extreme clarity and perfection in everything she undertook. Her photographs are among the most beautiful X-ray photographs of any substance ever taken." She published 37 scientific papers. What happened then? Well, here she is. Here's her lab. Picture taken shortly after her death. And one of the things that's interesting on this table, I don't know if you can really see, but on the table are cutouts for model building. And one of the things she became known for is her incredibly detailed and beautiful models of the tobacco mosaic virus. So in 1962, the Nobel Prize in Physiology or Medicine was awarded to Watson, Crick, and Wilkins. It's awarded to no more than 3 people, and it's not awarded posthumously. Wilkins, in his biography, continues to really not understand what he might have done wrong. And he, in his autobiography, which is the last one to appear, if you notice on the list I gave you, he's still trying to understand why he may have been portrayed as the bad guy, and comes off fairly defensively. He also makes a case that really he deserved the Nobel Prize because he really helped them fine-tune all the details of the structure of DNA after the fact. Crick made far-reaching discoveries of the fundamental mechanisms of genetic expression. And over the course of his career, or after the DNA, became quite a good colleague and friend to Rosalyn Franklin, ironically enough. Watson remains a controversial hero. But even he and Rosalind had a professional correspondence and addressed each other by their first names. He didn't use Rosie to her face. In 1968, Watson published the Double Helix, and it was so inflamed, the preprints of it so inflamed his colleagues. In 2 years before it was published, Wilkins wrote him saying, "Concerning Rosalind, is there any mention in your book that she died?" Right. So Wilkins and Crick found the book to be so inaccurate that they petitioned Harvard University Press. Watson at the time was a faculty member there, not to publish it. It's the only time the press has pulled a book. But, then the press agreed, but the book was published by Atheneum, and it's continued to print. It's a bestseller, selling millions of copies, and has been very favorably viewed. It's an exciting story. It's a big ego. Watson continues to be defensive and to make derogatory statements about Rosalind to this day, even at the Bolin lecture that he gave at the university a few years ago. DNA continues to be studied. Oh, I don't have the picture. Here it is. DNA continues to be studied But there's much about that molecule that we still don't understand. And Rosalind is becoming more well known. This image is from our latest biology textbook, and those books now include a picture of her and her famous photograph 51, but the highlight really is given of Watson and Crick. This is the Rosalyn Franklin building at the University of Cambridge, which is housing for graduate students. There's a medical school facility named after her. The laboratory where she and Wilkins worked has been named for the two of them, ironically enough. And so her, oh, I'm sorry, I have the wrong, excuse me. This is the image that's there. And so her story has not died, and she is being appreciated more and more for all of the significant contributions that she made. And then we can ask, what is the lesson for how science should be done? Obviously, this is not a model for how science should be done or a model for interdependence. Increasingly, modern science requires international collaboration and efforts that will bring people together to solve or to address world problems, including hunger and disease. Thank you.